Meet Zach. Zach's body is functioning normally. His cells are replicating in a precise and orderly fashion following a set of signals in the cell growth cycle. But today, one of Zach's cells is about to mutate, causing it to grow wildly out of control, ultimately leading to a deadly cancerous tumor in his prostate.
There are 4 stages of prostate cancer, starting with stage 1. In stage 1, the doctor identifies that there's a tumor within the prostate. When it comes to stage 4, the cells of the prostate kind of break up and travel throughout the body. So the cancer spreads throughout the body and it goes to either the bones, the lymph nodes, or other organs. For stages 1 through 3, the cancer is actually treatable through surgery, radiation, or hormone therapy. But the treatment options become so limited when the patient presents as stage 4 prostate cancer.
Especially with an African American men, they don't always present at stage 1. They usually present at later stages with more aggressive forms. So that is actually what's being presented in clinic. So how can we treat those stages in these cases? And so in my research, I'm hoping to, I don't want to say cure, but I want to provide them more treatment options.
So that's my good friend, Dr. Corey Grayson. Dr. Grayson is a chemical engineer working at Cornell University / Vanderbilt University, and she is studying prostate cancer, specifically how to stop the spread of cancer and eliminate tumor cells that travel through the bloodstream. Could you explain to us a little bit more of the process of what your research entails?
My research mainly focuses on studying prostate cancer. Normally with most cancers, the primary tumor is not the issue or the problem. It's usually the spread. So 90% of cancer-related deaths are due to the spread, or what we call metastasis, of cancer cells. So our work in this lab, we look at targeting specifically CTCs, and CTCs are circulating tumor cells that have dislodged from the primary tumor, and they enter into your blood circulation and then find other ways to invade and then metastasize or make smaller tumors in these organs.
It's the spread of tumor cells that leads to stage 4 cancer if it's not treated or caught in time. So the problem comes up is that tumor cells are really good at hiding from our white blood cells as they travel through our bloodstream to other parts of our body. Our white blood cells are essentially our immune system's defense team. So if our tumor cells are able to hide from our white blood cells, then the question comes up, how can we come up with a treatment that is able to eliminate these circulating tumor cells and stop the spread of cancer?
So we have developed a nanoparticle or a liposome, so it's very, very small. And we have decorated with 2 different proteins on it. One is E-selectin, I'll talk about later, and then TRAIL. So TRAIL is specific and special because it targets cancer cells specifically while sparing normal healthy cells. And so that makes it good as a cancer therapeutic.
All right, let's start off with the definition of liposome. So liposomes are these small spherical particles that we can't see with our eyes or with our naked eyes, and they have a similar structure as a bubble. So think of them as like a biological bubble.
In the lab, Dr. Grayson decorates the surface of the liposomes with 2 proteins, E-selectin and TRAIL. E-selectin is a protein that white blood cells recognize as friendly and connect the liposomes to the surface of the white blood cells. Having the liposomes on the surface of the white blood cells creates a sort of invisibility cloak. When a white blood cell approaches a tumor cell, the second protein, TRAIL, attaches the liposome to the surface of the tumor cell. Once the TRAIL protein is connected, the tumor cell performs programmed cell death.
The reason this invisibility method is considered a good treatment for prostate cancer is because it specifically targets tumor cells that are circulating through our bloodstream, and it doesn't attack any other fast-growing cells, such as our cells that cause your hair to grow.
What we did is we took these proteins, specifically TRAIL, and put them on liposomes. And once we put them in circulation with other cancer cells, we saw that they reduced cancer cell viability almost down to 90%. So it's basically killing them in circulation. And that's what we want.
So why was this observation or this conclusion that Dr. Grayson came to exciting or interesting for them? In the long run, when we're thinking about cancer treatments, we want to make sure that they're not going to interfere with the body's natural system, the way it works naturally. So the fact that this invisibility cloak method didn't interfere with the body's natural function, and it kills circulating tumor cells up to 90%, makes this a really exciting and potential solution to treat late-stage prostate cancer for men. That sounded way too exciting. That sounded way too exciting, but it's just still an exciting endeavor to know that we're at least one step forward to coming up with a new solution for such a challenging problem. This video was compact with a lot of information, and if you didn't take anything away or learn anything about prostate cancer, I hope you got to walk away with meeting a chemical engineer, seeing the type of research that they do, and also meet Dr. Corey Grayson, who is an awesome scientist, and get to add her to the list of cool scientists that you know. Thank you so much for tuning in, and I hope to catch you on the next episode of Future Doctors. Later, fam.